got some.
Got everything else you need?
I think so.
What is it? Oh, do you have a pointer?
Yeah.
Yeah?
He looks a little.
Dude, we were just saying he looks like he's locked in a basement. Like he's being held as a hospital or something. Hands tied behind his back. We had two terrorists who are about to size that off. It's gone on the video.
Jihad.
Yeah. Arms gone on the jihad. Yeah. An emergency jihad to Hawaii.
I don't know how to do that, sir. We're gonna have one. It's hot air, so just spew at you guys. Yeah. We do that. I'll tell you where.
Do you have any pictures from that field crew? For your first year at UCD?
No, I don't, but I think he might. He might, yeah, he might.
Oh, Mar might. That'd be awesome.
Yeah. I'm a little worried about that.
Yeah. I have pictures. I have pictures of the me, Ryan, and Desi crew, but I don't have pictures of.
Yeah.
I just saw it, but I didn't read it.
The Al photos are the one that I have. The Al.
Yeah.
That's one that I have.
You just like giving up trying to guess.
So he's been drinking.
I think we should, you know, we should have a little game. We should have people imitate Marm and say what he would be saying based on that photo.
Yeah, don't play the sound.
So just mute it, and you guys can.
I found this really good stuff in my cover, and I don't know what it is. I have this brilliant idea. What? And then when I woke up the next day, I didn't think my idea was so brilliant anymore. So I gave it to Will. That's a good way I can insult two people at the same time.
Alright, so we don't waste any time here. I'd like to get started.
Isn't Fred introducing you?
No.
I'm introducing myself.
That's why Marm's.
The whole deal.
Oh, it's a pre-recorded.
I think you
It's a
should play that
pre-recorded
video.
video.
Everybody's
Make sure it's loud enough too. Where's Marm?
He's on the screen.
He's stuck
Right
inside the
there.
computer.
We all knew it was a robot.
But he talks fast, you have to pay attention. If you play
You played it half a year.
If only we could do that in real life.
So I think we'll get started. Marm's not able to be here today, but he pre-recorded a video introduction for me. So I'm introducing Marm, introducing me. Now. So we'll play. I haven't seen this yet, so we'll go.
You haven't seen it?
I have not.
How much have you edited this?
I have the great pleasure of introducing today's speaker, Will Janousek. I'm sorry that I'm not able to be there with you in person, but I wanted to tell you a little bit about Will nonetheless. I first met Will in 2008 when he worked for me on my West Nile virus research project in the Washington, D.C. area. 4 qualities about Will were immediately apparent. First, Will is very bright and picked up all the skills in the project faster than anyone else on the crew. Second, Will has great judgment and he was always focused on the research at hand. Third, Will is a born leader with almost presidential-like qualities. Fourth, he's not afraid to lay down the law when he really needs to. And perhaps most importantly, really to lay it all out there. While it's true that he is experimenting with some potentially mind-altering substances while in DC, I really don't think that Will regrets any of his experiences. And while he was occasionally caught like a deer in the headlights, he always laughed it off with a beer with a friend. One thing I really like about Will is that he was never afraid to try out new ideas, be it fashion statements or really creative facial hair. Okay, enough fun. Honestly, Will has done a really nice master's thesis, and I'm honored that he's my first graduate student to finish his degree. He's worked on a number of different projects during his time at UCSC, including ecosystem services provided by bats by controlling insect pests of crops, the impacts of West Nile virus on birds in the field.
And that's all I've done. Well, I guess that's all the video. We will we'll move on from there and get to what I'm talking about, which which is this. This is what we're going to talk about today. So I'm really happy to be here and share with you some of the work that I've done over the past few years. Coming here in 2009, I was really excited to work with Marm and get into the West Nile virus system that I've worked with him in the past as a field crew leader and member prior to joining him here at UCSC. So today I'm going to talk to you a little bit about avian roosting behavior and how that can affect vector contact rates for hosts of West Nile virus. Okay. So it's been quite clear, and many studies have shown that the behavioral culture of hosts plays an important role in pathogen transmission, generally for directly transmitted diseases. Some good work done by Tim Tinker and Jim Estes in collaboration with other individuals has shown that prey choice and habitat preference has been shown to affect disease prevalence in southern sea otters. Aggregation behaviors in tadpoles has been correlating with chytrid fungal infection, as well as mating strategies and social organizations in many different mammal systems. I put koalas up there because they're a fairly communal animal and they're very promiscuous. They have lots of sexual partners, and chlamydia has actually become a big problem for koala populations in Australia. But what I'm interested in, what we're going to talk about today, is something slightly different, which is vector-borne disease systems, which is different than directly transmitted diseases where hosts contact each other. In these systems, you have a vector, generally a mosquito or a tick, some sort of arthropod biting vector, transmitting the pathogen to vertebrate hosts. And that transmission is generally directed through contact rates between the vectors and vertebrate hosts. And so contact rates are sort of main drivers of disease transmission in a vector-borne disease system. Now, when I started to think about this, what types of behaviors could hosts be doing that would influence their contact rates with vectors? When you go to the literature, the majority of studies that look at host behaviors focus on human host behaviors and diseases that directly affect humans, like malaria, dengue fever. So how people move around the environment, how people change the environment, are some of the things that people have studied, but very few people have studied the consequences of wildlife host behavior on vector-borne disease. And so what we'd like to really focus in on is what are the consequences of wildlife host behavior on things such as vector contact rates and fine-scale variation of vector abundance. And this is particularly important for diseases where we don't know a lot about the transmission process, such as West Nile virus, which is what we're going to be working with today. And West Nile virus has a wide range of implications. It causes conservation concern for certain species, such as this yellow-billed magpie here. It certainly has human health implications, causing encephalitis in humans, and an economic concern. People that have livestock or own horses have been directly impacted from West Nile virus. So to give you a little background into West Nile virus, it was first discovered in New York in 1999, just in a neighborhood of Brooklyn, and it was subsequently found in a few other states that same year. But within 1 year, it had been found all over the eastern seaboard, and by 2003, it had crossed the country, went up into Canada, down into Mexico. Oregon was late to the party. It didn't show up there until 2005. But it's a very dynamic disease and spread rapidly across the country. And the reason it was able to do that is because West Nile virus is maintained in a transmission cycle between Culex species of mosquitoes, this guy here, we also like to call him brown house mosquito, and avian hosts. And so birds being fairly mobile were able to spread the disease quickly, and they're thought through migration patterns and such that that's why it was able to spread across the country so quickly. So this is our general transmission cycle, but occasionally you have transmission to incidental hosts. We like to call them dead-end hosts. My roommates were found out on this one, but humans and other mammals are not don't replicate the virus well in their systems, so don't infect that many new mosquitoes. So primarily, we see the transmission process being between mosquitoes and avian hosts. So if we focus in back on the birds, we found over 200 species of birds have been shown to be infected with West Nile virus. And these are sort of just a collage of birds that we caught on the field projects in DC. However, one single species has been implicated as the most important species in West Nile virus transmission, and that's American robin. And for two reasons. American robins are really good at amplifying the virus once they're infected, meaning if they get infected, they are able to produce lots of virus particles, and then they can infect new mosquitoes. And mosquitoes that transmit West Nile virus also prefer to feed on American robins. And what I mean by that, we can look at this figure here, we'll discuss what I mean by preferential feeding. So on the top we have 5 different sites. Each bar is a site, and we have multiple colors here, and those correspond to different avian species. So on the y-axis we have avian abundance. You can see a lot of blue, and these big blue bars. It's all house sparrows, so they're fairly abundant at all of our sites. But what we don't see is a lot of red here. That's showing us that American robins are fairly uncommon at each of these 5 sites. Now, if we look at the blood meal data, though, here we have percent of QX blood meals, and we see a lot of red now. It shows us that this species of mosquito are feeding a lot on robins, even though they're fairly unabundant at our field sites. So it's kind of an interesting process that's happening. Why is a species of mosquito feeding on an avian host that's fairly unabundant? What's causing this process to happen? And really the answer is we don't quite know that. And so today I'm going to sort of dive into some possible reasons for this preferential feeding, and maybe convince you of that. So brings us to our first objective of this project, is to determine those mechanisms leading to preferential feeding by mosquitoes. And one of the things we wanted to look at was, how does mosquito abundance vary across different heights in the canopy? There's been a lot of studies that have been done that have shown that as you move here, like on the y-axis we have trap height, and from the ground to the top of a tree. We have the number of mosquitoes on the x-axis. And so there's been plenty of studies that have shown mosquito abundance increases as you move from the ground up to the top of the tree. On the flip side, if we think about avian roosting behavior, we think about roosting height. It's not something that's been well studied, but through natural history observations of species, we know that species that feed on the ground and breed on the ground probably are going to be roosting somewhere close to the ground, versus species that breed in the canopy and feed in the canopy will probably roosting there. So we expect to see some sort of variation in avian species on their roost height. So if we bring the mosquito data, what we know about mosquitoes back in, suggests to us that perhaps species like the bird species you see here, roosting higher in the canopy, will be exposed to more mosquitoes. Therefore, they might be fed on preferentially by those mosquitoes through contact rates with them. So that brings us to the first hypothesis, is that avian species roosting higher in the canopy will come in contact with more mosquitoes, and that is what will lead to the preferential feeding by the mosquitoes on those avian species. So the next part, we're looking at avian communal roosting behavior in West Nile virus. So there's been a few studies that have been done that have shown these large aggregations form in the fall, or not quite the fall, but the late summer. And they form these large communal roosts. And there's been quite a few studies that have shown that for reasons of information sharing, when food starts to become scarce, birds flock together so that they can learn from each other where they're feeding. But a lot of people took this from a disease standpoint when West Nile virus became pretty big and looked at robins, because they are the species that do this communal roosting behavior, and they suggested that communal roosts act to amplify transmission, act sort of as transmission hotspots. So this study in 2010, this Janousek study, showed that communal roost sites, this large gray bar, had more percent infected mosquitoes than non-communal roost sites, or sites that communal roosts did not exist at. However, another study that came out in 2012 said, well, we don't really see a spatial pattern. It seems that we find infected mosquitoes there, but not quite any. There's no quite strong difference between roosting sites and non-roosting sites. I think this is interesting, what these guys are starting to look at, but what Marvin and I started to talk about, and what we're more interested in, is what about the vector-to-host ratios at communal roost sites? So like in this study here, they found more infected mosquitoes at communal roosts. But if there is a lower amount of mosquitoes per bird, you still could have reduced transmission than if you have more infected mosquitoes and more mosquitoes per bird. In that case, we would expect more transmission going on. So really getting down to what the vector-host ratios are at roost sites is what we wanted to do. So we went to look at mosquito foraging behavior at communal roost sites. There's been a few studies that have shown across multiple types of taxa that as you move from small to large groups, you catch more mosquitoes, and there's a larger abundance of mosquitoes as you increase your group size. But we're not quite interested in just abundance data. We're really looking at, does the per capita number of mosquitoes increase with larger groups? And so that's that vector-host ratio, that's the number of mosquitoes per host that we're really interested in. So that brings our second hypothesis. We wanted to go look at communal roosts, and we hypothesized that mosquito abundance and vector-host ratios would be elevated at avian communal roost sites. We hypothesized this because we believe that these large groups of robins produce enough CO2. CO2 is what's attracting the mosquitoes to the host. So these large sort of beacons of CO2 would bring in mosquitoes from the surrounding area, and then that's why they're acting as transmission hotspots, because they're elevating the amount of mosquitoes at the communal roost sites. Now there's one more piece of the puzzle that I haven't discussed yet, and that's whether if you place a mosquito trap inside of a communal roost, could the presence of the host around that mosquito trap distract mosquitoes from your mosquito trap and bias your abundance estimates? So this cartoon here, we have a mosquito trap in this roost here, we have a mosquito trap that's kind of near birds but not really being affected by them. And so we wanted to ask the question, could you get some sort of bias in your mosquito estimates and then ultimately bias in your vector-host ratios? And there was a study done in 1983 experimenting with ungulate herds and reindeer, and they found that depending on where they placed their trap within the herd of reindeer, they caught more or less mosquitoes. And so traps placed in the center of the herd caught less mosquitoes. Traps placed outside on the perimeter of the herd or away from the herd caught quite a bit more mosquitoes. So we really wanted to think about that after we started to look at our mosquito abundance data that we collected at our field sites. So that's our second objective, was to determine whether the presence of hosts near a mosquito trap could affect trapping effectiveness. And we have no a priori hypotheses for this. We weren't sure if this was even occurring. We just decided to take a stab and see if this process was happening. No other studies, none of the disease studies that are going on, especially not for West Nile virus and anything that's doing mosquito abundance estimates, have thought about this in any of their studies yet. So it's sort of a new thing for us to put into the system. Okay, so where did we do the work? Well, we had 3 field sites in the greater Washington, D.C. area, sort of on an urban to rural gradient. One of our first field sites was in downtown Washington, D.C., at the Watergate Hotel right here, and then from this building where all the Nixon folks were stealing things. This is our field site. It was great working in downtown D.C., although you could get hassled by the cops quite often walking around with binoculars. There's in Saudi Arabian embassy here, the Egyptian embassy is here, so it's kind of tricky at times. So just outside of the district was our second field site. It was in a residential area, and it's just outlined here. Just a nice neighborhood to work at. One of our field houses was located there. And then our third field site was in Rock Creek Regional Park, just outside of Rockville, Maryland, and it was a pretty much forested site. So for American robins and northern cardinals, in order to get our birds to assess the roosting behavior, we had to catch them and put transmitters on them. But for American cardinals and northern cardinals and American robins, we started following those guys from the nest. We put transmitters on fledgling individuals, and this was part of a side project that we were looking at fledgling survivorship also. But today I'm just talking to you about this side of the project. So we did some nest searching. Emily here is looking at a cardinal nest using a mirror on the end of the pole. This is a robin nest, and this one is a northern cardinal nest. So after those birds were old enough to be transmitted, we took a few measurements, and we attached these radio transmitters onto them. We also were vaccinating some of the nestlings. That was part of the survivorship study that the results are still being analyzed for. So in total, we collected data on 7 species. We had to use misnets to collect individuals from these other 5 species here: house sparrows, European starlings, gray catbirds, tuft titmice, and mourning doves. Emily is holding a northern cardinal there. And so once we had all of our birds and we were able to put transmitters on all of them, we could go out and track them. But since we're studying roosting behavior, we had to go out at night. We called that our nocturnal telemetry. And I had to make my field crew start to wear these reflective vests. Emily was not very happy about that at first. In part because when you're walking around downtown DC with radio telemetry equipment, you do get hassled by the cops. This is a photo taken from a trail cam that we had to put out. The police had came because they thought we had put a bomb against, or strapped a bomb to the tree. But it was still had a lot of fun. And so once we used the radio transmitters to locate our birds, we used thermal imaging cameras. This isn't a photo of our birds, but this is to give you an idea of on the fine scale setting, when you got into the vegetation, we use the thermal imaging cameras to pinpoint our birds, and then we can measure the roosting height, and then also count the number of birds they were with. This is actually a photo from one of the thermal imaging cameras. It's quite blurry, but on the lower quality settings, the warmer an object was, the darker it would show up, and it was really easy then to use that setting to count individual birds within a tree. Because I think these are robins roosting in some bamboo. So briefly, I just want to discuss, I don't go into detail in my figures about my analytical methods, but just to give you an idea of what I did, we used generalized linear mixed effects models to do both our roosting height analyses and our roost aggregation size analyses and all of our mosquito abundance estimate data we did. We analyzed using mixed effects models. This is all done in the LME4 package in R. So if you have questions about my my analyses in those models, we can definitely talk about that at the top. So to return to this, just to refresh, to return to this figure, we expected to see an increase in the number of mosquitoes as we tracked higher in the canopy versus lower the canopy. To do that, we first set out traps along these string trap lines where we had 3 different traps in the same location, using mosquito traps like this here with the CO2 cooler. It was a cooler full of CO2 dry ice that acted as our bait. And we've seen that we've been doing mosquito trapping at all these sites for a long time. The study started back in 2004, so we have quite a bit of back data to show that we do see a pattern of catching more mosquitoes at high traps than low traps, but we wanted to confirm this pattern occurring while we were doing our roosting study. So we did this. Just wanted to give you I guess a better understanding of how the traps work. There's a fan here, and it blows into the catch basin, and so a mosquito comes along looking for the CO2 that's coming out of this cooler, and then finds its way into the trap. Okay, so what did we find? Well, we found that pattern that we expected to see. We saw that on the y-axis here we have mosquitoes per trap night, and we have our 3 different trap heights. And a pretty consistent pattern across all locations that we caught significantly more mosquitoes at high traps than at low traps. So then to return to what we saw with bird species, when we expected that those bird species roosting higher in the canopy, we expected to see some sort of variation, and that those birds roosting higher in the canopy may be contacting more mosquitoes. We found some variation in roosting height. So we have our 7 species here on the x-axis, and roost height in meters on the y-axis. Now these dots that each species has is an actual observation, and the darker the dot, the more observations we have for that specific roost height. These aren't typical box plots. What I've done here is the mean observation for a species is this bold horizontal bar. The boxes around those bars are the standard errors, and then the hash marks are the 95% confidence intervals around the mean. So we kind of changed this up a little bit from a standard box plot, but I think it tells a little bit more about what's happening. So we see that titmice roosted significantly higher than all other species. These letters designate significant differences from each other. Not a lot of variation going on here. Robins and house sparrows roosted at significantly different heights from each other. But again, we want to ask the question now, bringing in the mosquito data and the roosting height data, and ask, do mosquitoes feed preferentially on species roosting higher than canopy, like titmice? But before we can do that, we have to calculate these mosquito feeding preferences. So to give you an idea of how we do that, we take the fraction of total blood meals from a given host, we divide that by the ratio of the density of the host, divided by total avian density. And that gives you something like this. So I have 2 species here. On the y-axis, we have mosquito feeding preference. We have our 2 species, house sparrows and robins. A feeding preference over 1 or greater than 1 means that a species is being fed on more often than expected. Under 1 is being fed on less often than expected. So from our field sites in DC, we find that robins are fed on almost 3 times more often than you would expect based upon their abundance. And for house sparrows, they're actually fed upon 8 times less often than you would expect based upon their abundance. So then we wanted to take in all of the data we have for all the species and we put it in a linear model here. So what we're going to have is roosting height on the y-axis and mosquito feeding preference on the x-axis. And what we found when we run the linear model is that feeding preference increased with host roosting height. And I've centered these photos over the points that they represent, but just to give you an idea of which species are falling where on the graph. If you're an astute observer, you notice that I did do roosting height analysis for 7 different species, and 2 of them are not on here. We excluded them before doing this analysis. Starlings we didn't include because this is focusing on hatchier individual or hatchier's roosting height, and so for our starling individuals, we have pretty much all adult individuals, and so we didn't really include them. And then also titmice, we unfortunately don't have a reliable estimate for their feeding preference for the sites that we did roosting height data at. But from me looking at the data and looking at sort of where starlings and titmice would fit if we had a little bit better data, titmice fall right in this area, and starlings would fall right down in here. So just to give you an idea of where those guys were. But we thought this pattern was pretty neat, and seeing this increase in feeding preference with roosting height. So what are some other implications of roosting height? Well, if mosquito abundance can be 6 times larger at high traps, which is what we showed in our stream trap estimates, then we would expect birds roosting high in the canopy to come in contact with more mosquitoes, and by doing so, this could lead to intensified transmission, and intensified transmission for some species can mean population declines, and titmice and robins have both shown population declines since the introduction of West Nile virus in North America, and these were the 2 species that roosted the highest. So moving on now, we're going to get to our second hypothesis here and looking at vector-host ratios and mosquito-host seeking abundance at communal roost sites. Now, so as we're going out there, we're measuring the roosting height of individuals, we're finding them with the thermal imaging cameras. We're able to count the number of birds that are roosting around them. For some species like robins, starlings, house sparrows, we expected to see those birds form communal roosts, although we did not know whether or not those species were actually forming roosts at our sites. Other studies in Connecticut and Chicago have found those species forming communal roosts in the late summer. So we expected to see this. We decided to start counting the number of birds that species are roosting with. But for 4 species, these 4 here, we found no seasonal pattern for changing in roost size. So we have roost size on the y-axis, and then we have our 4 species here. Again, these are the same box plots with the mean, the standard errors, and the confidence intervals. So now we're gonna switch scales here. We're going to turn this on a log scale and introduce those 3 other species that we expected to see some sort of communal roosting behavior. So this is data from the first week of our study. So these are the roosting size values for these 3 species, house sparrows, starlings, and American robins during the first week. And you can see that they don't vary that much for 4 other species. But when we add in their data from the end of the study, again, we're still on the log scale here, we see a pretty marked change in roost size for each of our species. So now we have the last week, the mean values for the last week for each of these 3 species. And since robins are particularly important in transmission, or believed to be, we wanted to sort of focus in on those communal roosts of robins. To give you an idea of just the actual robin data, each one of these black marks are an observation for a robin species where it's given roost height, or roost size, which is here on the y-axis, across the length of our study. And I've highlighted a single individual in red. Each one of these is from the same individual as he's progressed, or she has progressed through time, moving to these large communal roosts, which are sort of these clumps here. I've jittered the points slightly just to make it easier to see. But we saw this general pattern of robins moving out of our study areas, joining smaller groups, and those small groups then forming into large communal roosts. And some of them were quite far from our study areas. This one here this yelled out here was about 4 miles away, and then these other guys we found during the course of our study. This is downtown DC at the National Mall over here. This is the Lincoln Memorial. This is in Arlington. We had, I think we had a communal roost in Arlington Cemetery, but we could never get in there to confirm that. And this was from our residential site. We found a few roosts around there. This one was in a parking lot of a metro station. There's large roost that had probably about 10,000 birds. It was a sort of a mixed flock of house sparrows, starlings, robins, a bunch of other things. So what we wanted to ask was this communal roost behavior affect mosquito abundance and effective host ratios? So we took our Tacoma Park site. The site turned it a little bit on the axis just to make it easier to see. We have 2 communal roost sites here in blue, and we have our 2 non-communal roost sites, or sites that were not used as communal roost. Now, there were probably birds roosting there individually, not in pairs, but we wanted to use those sites as our non-roosting sites. And so from there, we put out between 2 and 6 mosquito traps, depending on the size of the site, and we ran those traps for a period of 5 trapping periods over 4 to 5 days. And again, that's sort of the trap setup that we were using. We expected to see these communal roosts having more mosquitoes, right? These large aggregations of birds pulling in mosquitoes from the surrounding area. Oh, I guess I have photos of the sites. I didn't forgot about this. So yeah, these are non-communal roost sites. There's a park here in this sort of stream corridor. Has a bunch of trees, and then another park with another stream corridor. And so we have our roosting sites are a little bit similar. This is a big bamboo patch behind a house, and we saw, I think on the largest point, there's about 530 robins in here, and then this site was in an apartment complex, and I think at maximum it was at almost 3,000 birds, American robins there. So those were our 2 communal roost sites. And to give you an idea of the vegetation inside there, it's pretty dense, thick vegetation. We thought it was pretty good mosquito habitat. The other one was a big bamboo stand like this. And so we were really primed and ready to say, we were going to catch a lot of mosquitoes at these communal roost sites. But what we actually ended up finding was we caught significantly fewer mosquitoes at communal roost sites than at our non-roosting sites. So here we have mosquitoes per track night on the y-axis, and we have our non-communal roost sites, our communal roost sites here in blue. And we found that we actually caught significantly more mosquitoes at our communal roost sites than at non-roost sites. Just to give you an idea of sort of what was going on. There's lots of birds around our traps in our non-communal sites. There may have been a bird or 2, but it wasn't a huge aggregation like we had observed at the other sites. When we compare, what's more important, what we're really looking at is the vector-host ratios, which again is the number of mosquitoes per bird. And they're insanely small at communal roost sites. It's like 0.001 mosquito per robin per night. It's very small, 2,500 times lower at communal roost sites. We thought this was interesting, but then if we remember back to our second objective, right, it was to determine whether the presence of hosts near a mosquito trap really lowered trapping effectiveness. We thought, if we go back to this, we thought if we put a trap out there and we had all these birds around it, could those birds be distracting the mosquitoes from the trap itself and causing us to have low bias mosquito estimates versus a trap that doesn't really have anything affecting it. So what we did was we set up a simulated roost experiment where we have a normal trap here set up by itself. It's a baited trap with a CO2 cooler. So that just to replicate what we do at all of our sites, it's a normal trap setup. And then we had an artificial roost setup. We put 4 additional coolers around the normal trap, and they were spaced about ½ meter to a meter away. And we sort of set them up in a spiral staircase pattern to provide some sort of even distribution of the CO2 coming out of those coolers. And really, we wanted to replicate it as closely as we could to what we saw at our mosquito traps inside communal roosts. We often saw robins even roosting on top of the traps at our communal roost sites because the densities of birds was so high. To tell you a little bit more about this, we measured the sublimation rate of the dry ice in these coolers and then determined the amount of liters of CO2 leaving the cooler per hour. And then we compared that to the field metabolic rate for robins. We used field metabolic rate for robins instead of basal metabolic rate because often, even at 3 o'clock in the morning visiting these communal roosts, we had birds flying from perch to perch and being very vocal. So we decided to use field metabolic rate. We thought that was a little closer. And it turns out that this artificial roost that we've created, not including our normal trap, but these additional coolers added to surround the trap, was roughly about 370 birds. And we thought that was a good estimate for the number of birds within 10 meters in close proximity of our mosquito traps in our community sites. And so we replicated that experiment across 4 paired locations with 32 trap nights. And we found, here we have mosquitoes per trap night. We have 2 different years, and on the red bars are our normal trap setup, and the blue bars are our artificial trap setup. We found on both years that we did the study, we found significantly fewer mosquitoes in our artificial roost setup than in trap ran normally. So we thought that was pretty interesting. We said, well, this could be happening in our actual communal roost. So now I've summed that experimental data between both years, just to remind you that we caught less in an artificial roost setup. On average, we caught 57% less mosquitoes in the artificial roost than from a trap by itself. And so we wanted to ask the question, can we correct abundance estimates at our actual communal roost sites? What would that look like? Would we then have a huge amount of mosquitoes like we expected to find? So what we did was we took the number of birds at a communal roost from our counts that we did, divided by the number of birds that our artificial roost setup replicated, and then multiply that by the percent reduction that we saw here. We just used the mean percent reduction. And this gave us our expected reduction at our actual communal roost sites. So if we bring that data back, now again here we have mosquitoes per trap night on the y-axis. We have our 2 sites, our non-communal roost sites and our communal roost sites. And then this is our adjusted communal roost site value for the number of mosquitoes. We no longer see a significant difference between communal roost and non-communal roost sites for total abundance. But what about the vector host ratios? And so what we find, we still saw that there was still so many birds there that our vector host ratios were still over 850 times lower in a communal roost than outside of it. So why is this important? Well, we know that social grouping can confer some sort of fitness benefits. Typically thought of from protection of predators, or like I said earlier, sharing foraging information. But we think robins may actually be reducing the risk of being bitten by mosquitoes by forming these large communal groups. And if you lower the per capita feeding on robins at communal roost sites, this could lead to reduced disease transmission. So just to summarize the talk, we saw a mosquito feeding preference to increase with roosting height. And we found mosquito abundance was significantly lower at communal roosts. Although we performed the simulated roost experiment, and that showed that hosts near a mosquito trap could alter or reduce the trapping effectiveness of a trap. That's something that we are, in the manuscript that we're writing up, we're really focusing on and suggesting to people that are doing future studies to take into account where and how they put a mosquito trap if they're trying to determine mosquito abundance at communal roost sites. So after correcting for that reduced trapping effect, we still saw vector host ratios almost 850 times lower at a communal roost, which for us means robins are benefiting from roosting communally by lowering their contact rates with vectors. So I'd like to thank my advisor, Mark Patrick. I'm really happy that he was able to produce a little video of me. That's very kind. And my co-committee members, Tim Tinker and Bruce Lyon, for their help in the thesis writing process. I really appreciate that. We had a lot of funding from National Science Foundation and NIH that helped run this project, and also summer research funds from our department. I always have to thank your lab, and my lab has been very supportive, and all the stuff that I've done here, and most of all my family who are here today. They flew here from Kansas to make sure I didn't fail. So with that, I'll take any questions that you may have.
Why does CO2 trap mosquitoes?
Well, so CO2 is one of the main attractants that a mosquito is using when it's trying to find itself, since we all respirate, right? We all exhale that CO2, and so that's sort of the first level of attraction that mosquitoes have. And then after that, once they get near a host, they'll use some thermal, this is here imaging, but some thermal reception to find particularly where to bite the host. But yeah, CO2 is the main attractant.
Yeah, I didn't do the math in my head, but if you had corrected for the artificial roost experiment on your non-communal roost, was that just a totally negligible difference?
So we didn't do it for our non-communal roosts. We suspect we were really only looking for the effect in a large aggregation of birds, and using the thermal imaging cameras at our non-communal roosting sites, we didn't see any birds around our traps. We didn't suspect that there was any distraction effect going on there.
Even if there were like 10 birds?
Even if there were 10 birds there, we use the thermal imaging cameras to look, and within 10 meters of the traps is what we expect 10 meters to be about the distance that a mosquito would be distracted by. And so we use those cameras to sort of make sure that we were having that bias also to keep it pretty even.
I was wondering if, like, how negative is the impact of West Nile on robins or other birds? Because that would be a negative effect that would require, like, for the hypothesis that they're communal roosting to avoid.
Sure. We're not suggesting that they are forming communal groups to avoid being bit by mosquitoes, but by doing so, they're actually benefiting from that formation. There's a lot of thought been into it for forming these communal groups for foraging, sharing foraging information and stuff like that. But I think once those roosts are forming, this has been a behavior that's been happening for a long time, even before West Nile virus was introduced. That once you're in a roost, being inside of a roost may be better.
I want to follow up on that, but again, the word benefit, we might be using it differently, but is there a cost to being affected to the birds?
Well, beyond mortality, I think.
No, I mean, is that what it is? Do they always die if they're infected?
Not always, no. So when you're infected, typically you have, after infection, you have about 7 days when you're infected. For robins, we're talking about. They tend to not be able to forage as well. They're little. When we watch them in the field, you can sort of sometimes tell which ones are infected. I don't think there's a real overall cost other than not being able to forage well. If you're nesting at the time, you might not be able to get food for those nestlings. But when you're in a large aggregation of birds and you're not nesting anymore and you're mainly concerned with feeding and getting ready to migrate, like a lot of robins are short distance migrants, being infected at that time could mean not being able to put on enough fat reserves to make that migration. So yeah, I think there are definitely some costs involved. Yeah, Joe?
I thought the part about the distraction factors was really interesting. I'm wondering what you think about, does anyone use non-baited traps? Because I haven't quite thought this through, but I would think that'd be an important control.
It depends on what you're trying to estimate. There's if you're trying to estimate overall abundance, we use baited traps essentially to pull in as many mosquitoes as you possibly can. Some, instead of CO2, some use pheromone traps. Those tend to be a little less effective. There are non-baited traps, but those are usually looking for mosquitoes that have already fed, and so trying to catch mosquitoes that have taken a blood meal, so that we can do some of those mosquito feeding stuff.
I guess there are insect traps that are stuck in flyways or whatever, because insects are down and then towards the light.
Yeah.
But I guess it would be hard. It's hard to think of one for mosquitoes.
Yeah, well, some of those CO2 baited traps also have a light on them too to add it to that, but the ones we use just the CO2.
I don't know, maybe there are enough bird nerds running around with binoculars to answer this, but is there any indication that average roost size has increased since early 2000s?
The actual size of the roost?
Yeah, that more robins are tending to roost in large groups than
The robin's size, roost size, is fairly spatially variable. I know the studies that have been done in Chicago have robin roosts in the order of like 10 to 15,000. In our area, we only saw them get up to 2,000 to 3,000. In Connecticut, they had them up to 5,000. So I think it might be sort of location dependent. And these studies go back to the early stages, just kind of post introduction of West Nile virus. So I would suspect that they probably haven't changed over time. If not, you might not have enough time has might not have elapsed to really observe the change.
Yeah, I thought that a lot of the questions that you brought up about CO2 and exactly the placement of things could affect feeding rates was really interesting. And one of the things that I was kind of thinking about is going back to the earlier portion of the talk where you're talking about different species. Is there a way to estimate how much CO2, like, do they vary greatly in the amount of CO2 they produce?
They do,
Would
because
it be enough?
they all have varying respiration rates, right? So we use the field metabolic rate for robins because we were trying to emulate a robin roost, but if you were interested to say, if you wanted to select for other individuals, like maybe we wanted to do it for house sparrow roost or starling roost that we saw these other species, yeah, we certainly could do that, and you would have to
You calculate a different amount.
another way, but it would be, it's a pretty simple.
Do you see, like, do mosquitoes respond in, I mean, does there have to be a big change in the amount of CO2 in order for mosquitoes to act, be attracted in a different pattern, or is it, does that, do you see what I'm saying?
Kind of.
How much of a difference in CO2 production, like either a trap or a roost and grouping of birds, would there have to be in order for the attraction of mosquitoes to be different?
Sure. I'm not sure if I can answer that directly, but so with our mosquito traps that we put out, and the way that everyone does this across the board is you fill a cooler full of dry ice, and no one really measured how much you're putting in there.
Got it.
And so when we measured it at full capacity, it was like 1,000 birds, depending on which metabolic rate you would use per hour. And so we had to, knock that down quite a bit to get it to be like that. So I think that'd be interesting to look at. The way that people are doing it now is pretty much overloading the environment with CO2 just to sort of attract as many mosquitoes, and not doing it from the other end of and see how little we can put out and see how many we can get in that one. Yeah, that'd be a whole other.
Totally. Thank you.
This is a comment on that, though, just from your committee meeting, but doesn't it look like it's a very local spatial scale? Because what the results suggest is even these massive roosts don't have more mosquitoes.
Yeah, I think
so it's not so it's going to be a very local spatial scale. Otherwise, you'd think if they're moving those long distances, we should get a huge pulse around these roosts, and we're not. So that suggests that you're affecting their local movements and not their global movements.
Yeah, I mean, there's some data that shows that mosquitoes have been known to move up to 1,000 meters in the course of an evening, whether that was host-seeking or just sort of random walk. But I think it has a very local effect. Yeah.
If that's the case, would it be possible to correct each estimate from your photos based on the number of birds and the immediate vicinity of each trap, rather than having one degree of correction?
Sure. Yeah. So this was our first stab at it. It's sort of what is the effect, and maybe how do we adjust for it? You'd have to have photos first to do that. I have like that one photo that we took, but I think it's something certainly interesting to think about, and certainly stuff that Marm and I are talking about now as we continue to measure these mosquito abundance out in DC is how we want to go about doing it correctly, as we're trying to do now, and propose to others to do correctly. I think we need some better examples of how to do it correctly before we start going around sort of saying that this is the new way to do things. Any other questions?
A couple of questions about the roost height. So first of all, you show this relationship between higher height and increased susceptibility. So why isn't the titmouse getting slammed with these things, right?
So from some sites, it is fed upon preferentially. It's fed upon more often than you would expect. For the field sites that we collected roosting height data at, we don't have good blood meal estimates for that species, so we can't make an accurate estimate for its feeding preference, so we didn't include them in that model. From our other field sites, we saw them being fed upon almost twice as often as you would expect. We have some forested sites closer to downtown DC where mosquito abundance is a little higher, and so there's a little more mosquitoes there, and you can get more blood meals. when you have low mosquito abundance, you also get less, you find less blood nets because you actually have to catch those mosquitoes after they feed. So when you already have few starting out, it's hard to catch more than have.
And then, so relationships within and among species. Do robins, does the height of their roost, is it influenced by the presence of and abundance of other species?
The height of the communal roost?
Yeah, do the bird species influence one another's roosting height?
The bird species or the individuals within the roost?
No, the bird species. So a tree with fewer of the other species, will the robins roost lower in the tree?
I would suspect, from our observations at communal roost, a lot of the variability in robins' roosting height came from our observations of them at communal roost, where we had some roosting almost on the ground because these roosts were so dense, and we had other individuals roosting at the very top of the tree. So in those areas, I think those robins probably pushed out the other birds that were going to be roosting there, and they sort of took over and went wherever they wanted to go, or where they had space to go. So I think in the communal roost, you wouldn't have any sort of effect like that.
So what about like density dependence? Do you have, with a smaller group of robins, will they mostly be lower in the tree, but then as you increase the roost size, does that move them up into the tree where they become more susceptible?
I don't know. I don't think I have an answer for that. Yeah, I think from the early data from parts of our study before they were not roosting communally, they often roosted near their nest height level, which is like 5 to 6 meters.
Not
right?
very high.
What's that?
Not very high.
Not very high, but which is sort of near their average. But then as they joined these communal groups, then the variability of that shot up, and you had them roosting at the, as low as they can be, and then also as high as they can be. So I think kind of went in both directions. Yeah.
I don't know anything about robins. Would an individual in one large aggregation sometimes be kind of go off by itself or going smaller groups, like are they kind of in groups, or is it individual?
Yeah, it's a very dynamic process. There's since we had our birds tagged, we were able to watch them move between communal roosts that we had set up. So at our residential field site, those communal roosts were fairly close together. I mean, there was like about 1 mile, 2 miles apart from the farthest ones. But we'd often have birds switch roosts, then go back to the same roost, or leave the area completely, new birds coming in.
So then it's really not for lower your chances of being infected as a bird. You want to stay in the large roost as long as possible or as long as possible.
If you were perceiving that you were getting bitten less often in a communal roost, yeah, you would want to be in the largest roost. And then also from our artificial roost experiment, we had those traps located at the center of the roost, right? And so that's where we had our lowest vector-host ratios. But you can imagine if you were on the outside of a roost where the host densities were a little lower, your contact rates might be elevated. So not only being in a larger roost, but also in the middle of the roost.
Well, and I wonder how they're all not infected because at some point you're going to be on the periphery of the group where you're going to find out by yourself and a small group, and then if you then join a large group again,
Sure. Well, the reason why we think they're not all infected is because the vector-host ratios are so low. It's just so diluted down that almost zero chance of bumping into a mosquito, let alone an infection, since not all mosquitoes are infected. Good? Thank you.
There's gonna be a little reception, some drinks, stuff.